Hi everyone, this is Maris Haddad with my final project, Decoding Cancer Types with Microbiome Data. I'm just going to go through a quick overview of what I did. So, the importance of cancer in the microbiome is that there's an interplay in the microenvironment where a tumor is between the human immune system, the tumor, the healthy cells, and the microbiome. So what I'm looking at is potentially predicting cancer type from the microbiome sequencing data. So first, I did some data pre-processing, rarefaction, log transformation, and centering and scaling. And I did this in R and then saved the data and imported it into Python, where I split it into the test, validation, and training sets. My first model was a feed-forward neural network, and playing around with this, I found that I really needed some dropout layers to prevent overfitting. And then I got a really good classification accuracy of about 82%. Now, looking at a different approach, using an autoencoder to classify the different cancer types based on the microbiome sequencing data, I found that this has similar accuracy, about 83%, but there's less overfitting even than the neural network with the dropout layers. And this is interesting as well because the autoencoder reduces the dimensionality similar to how a PCA reduces dimensionality, in concept at least. This is important because in microbiome it's very standard for us to perform PCAs to better understand our sequencing data. So I visualized the latent space and put it next to a central log ratio PCA, which is pretty standard in the field. And you can see that the cancer types cluster together very significantly in the autoencoder latent space, but they don't in the PCA principal components 1 and 2. So essentially, what this means is that the autoencoder is capturing features in the dataset that the PCA is not, and I hypothesize this could be because the PCA is based on a linear approach, whereas the autoencoder can capture nonlinear information. What this means overall is that autoencoders could potentially help us to uncover significant findings about how the microbiome interacts with cancer sites that traditional approaches like PCA would not be able to do. And so I took another step and I tried a data imputation with the autoencoder just to make sure that it was really learning these interesting features that the PCA was not able to capture. And I found that there was clustering in the imputed data. And so as you can see in this data set, 10% of the values were masked and then imputed. And again, we see clustering. So this is a good sign that the autoencoder is actually learning features in the data set that the PCA isn't necessarily learning, and therefore it's able to classify the type of cancer based on the microbiome data from the site accurately. So in conclusion, the autoencoders offer an interesting alternative, or perhaps additional step to traditional approaches in microbiome, because they may be able to explain features that could end up being clinically relevant and could impact the treatment of patients in the future in a very positive way. And in terms of future work, I would like to investigate the biological relevance of these key features, elucidate perhaps more information about what they could be. And I would also like to apply this approach of the autoencoder to data regarding survival time. So as the cancer progresses, do we see a change in the profile of the microbiome from the cancer site and from the blood tests, similar to how we see them here? And that is in such a way that we could, instead of classifying the cancer type, perhaps determine how much longer the survival time is of the patient based, again, on the microbiome. So thank you for listening. Overall, I found this to be a very important project, and if you're interested, then check out my 15-minute long video. Hope to see you there.